Hello, my name is Mr. Reif, and today we are going to do a lab about cellular division. Last week we talked about cell size. Today we're going to do a lab that involves mitosis, which would lead into meiosis eventually. First, we would take care of business with our daily 5 questions. The first question is, explain how your lab shows that small cells are more efficient than large cells. I would call on a student, have them explain their answer, and if they don't know, have somebody else help them out, call them by name. We just would talk about how the efficiency deals with nutrient and waste removal across the cellular membrane to make things more efficient. The second question is, list 2 problems that a large cell would have. I would once again call on 2 students, one to give an example of each. We would take 5 minutes to do our daily 5. Then we would move into the mitosis introduction, which would follow, if time would allow, a video to give them an introductory. Before we got into the lab portion, I would show them this quick little demonstration of a strawberry. It kind of gives you the prophase, metaphase, anaphase, telophase as a joke. Then we would talk about the entire cell cycle. You have interphase, G1, S phase, G2, where we would cover that in a video. Then I would show my students that on the board you would leave this up on the projector and have them get into pairs, and they would take a cardboard plate. In our instance, we're going to have just a white plastic plate. We'll need one of those for each group. For today, I have zip ties colored to represent homologous chromosomes, sister chromatids. They're going to make chromosomes out of it. They would have beads where they would put the bead on, and then it would split in the pairs. And then they would need their notebook, 3-ring binder. We would use yarn in other cases. And then I would have a cup at each station with all the supplies to represent all of the portions of a cell. They would need to have the spindle fibers, the centrosomes, centromere on a chromosome, which connect the two, and then they'd have to have as many chromosomes as they'd like. They're also going to need a phone, some scissors, various materials that they want to represent in their cell, and then we'll get into the lab where I'd leave this on the board, as I already mentioned, where chromosomes condense and you can start to see the nucleus, nuclear envelope start to disappear. What we would do is they would line their chromosomes all throughout the plate. Doesn't matter the orientation, just as long as the colors are the same together. And then they would take a picture of prophase, and I'd be demonstrating this in the front of class. Then we would move on to anaphase, metaphase, where the chromosomes line up along the middle. Spindle fibers would start to attach to the centromere. The spindle fibers would attach to the centromeres from the centrosomes, and then I would have other materials for them to take pictures of that, and then they would take another picture. We're going to make a video out of this. After metaphase, we have anaphase. Anaphase is where you start to see the sister chromatids start to pull apart, where the colors actually help distinguish the chromosomes and how they're separating to form 2 new cells, nuclei. Then we would take a picture of this. We would move into telophase, where we start to see 2 new cells begin to form. You start to see the spindle fibers break down. Take a picture, and then we move on to cytokinesis, where we have 2 new nuclei. I would give the students about 30 minutes to do this project in total length, and then afterward, or during that time, I would walk around. I would say, okay, right now you should be at prophase. Give them 10 minutes. At this time you should be at metaphase. 10 more minutes, or however long they take. At this time you should be at anaphase, telophase, and I would allow them to take pictures of this on their cell phone so that way they have a reference guide right there with them. So after they go through all that, I would demonstrate all that, show them, they would do all this, and then I'd be walking around making sure that, and they have to get my approval before they work on the next phase. Then as a class, for the last 5 minutes, we'd come back and we would discuss what they saw between prophase, metaphase, anaphase, telophase, and cytokinesis. They'd have to describe what's happening in each prophase. You have the chromosomes condensing. You've got metaphase, where they line up along the middle M, metaphase. Anaphase, the sister chromatids start to separate, and then telophase and cytokinesis. I would ask student groups to tell me what they actually saw, and then this would be the part where we talk about what we want to do with the video portion. I want them to commentate what they're seeing, depict or tell us what their depictions are. They can get creative with yarn, noodles, cereal, you name it. They can do whatever they want. They just have to be creative and follow these slides. If there's time after that, we would do a Kahoot, where it would test their knowledge on the video and their prior knowledge, because we would have worked on mitosis the day before. This would be a good review, and